Dear students, for understanding the different aspects of any metabolic pathway, it is very important that in addition to learning about the reactant and products which are involved in that pathway and the enzymes which are involved in that pathway, you also learn about how that pathway is regulated, because this regulation of a pathway is highly important for the physiological balancing, physiological maintenance of the overall cellular environment. So, glycolysis is not an exception, actually it is one of the most important biochemical pathway, and hence its regulation is of utmost importance. There are three reactions which are of most importance when we are talking about the regulation of glycolysis. Glucose 6-phosphate formation, the very first reaction of glycolysis, fructose 1,6-biphosphate formation, and pyruvate formation. The glucose 6 formation you have already gone through it, but keep in mind that depending upon the cellular environment, different enzymes are involved in this reaction. It is catalyzed by hexokinase isoenzymes 1 to 3 in most of the tissues. However, in liver and pancreatic beta cells, it is catalyzed by hexokinase 4. आइसोफॉर्म्स है, मुख्तलिफ एक ही जीन की ये प्रोडक्ट्स होती हैं, तो ये मुख्तलिफ सेल्स में मुख्तलिफ तरीके से एक्ट करती हैं. एक्ट मुख्तलिफ तरीके से मुराद, I mean that's why they are different. I mean they, of course the final product is same, but in terms of the concentration which is involved, in terms of the timing which is involved, they may differ a little bit. The hexokinase 1 and 3 are inhibited by the product of this reaction, which is glucose 6-phosphate. That is what we say the feedback mechanism, the more accurately the negative feedback. If more the product is being produced, it will definitely halt the reaction. Then a glucose kinase is not directly regulated by glucose 6-phosphate. Hexokinase होते थे इससे, hexokinase were regulated by the product, but not this case. This is the case with glucose 6-phosphate. The glucose not the case with glucose kinase. Here the glucose kinase is regulated by fructose 6-phosphate rather. So this is a little bit difference between how the two types of enzymes, that is hexokinase and the glucose kinases, behave. Then again we have talked with real focus on this, that how and why the fructose 1,6-biphosphorylation is perhaps the most important regulatory step, because here the presence of different moieties regulate the whole process of glycolysis. For instance, if ATP is more, of course, then the reaction of the glycolysis will be halted, because we already have so much ATP. If there is so much AMP, then you need more ATP. The reaction will be then moving towards the formation of pyruvate at the end. Then also keep in mind that as compared to ATP, fructose 2,6-biphosphate is even bigger regulator of this reaction and of glycolysis. Even if ATP, let's say you have less ATP, but fructose 2,6-biphosphate is there, it will still move the reaction to occur in the forward direction. And finally, the regulation of the pyruvate formation. You know, the pyruvate is formed in the last reaction of glycolysis, when phosphoenol pyruvate gives rise to pyruvate, and energy is produced in this reaction. The enzyme here is pyruvate kinase. Keep in mind that fructose 1,6-biphosphate here regulate the activity of pyruvate kinase positively in what we say feed forward strategy. We have been learning, you must know about the feedback mechanism, but there is also feed forward, that is in which a product of a previous reaction control the production of a product of a succeeding reaction, or of 2 or 3 reaction, reaction which is far away, that are by the step of 2 or 3 reactions. So that's feed forward mechanism. So in this way, fructose 1,6 biphosphate formation leads to the pyruvate formation. More fructose 1,6 biphosphate, more will be the pyruvate formation. But in case you have a starvation, you are facing starvation, and you then need more and more glucose, what will happen, that the instead of glycolysis, instead of the formation of pyruvate by pyruvate kinase, the reaction will move in the reverse towards making more and more glucose. That is what we say the gluconeogenesis. So it happens in liver, and it happens in other cells where the deficiency of glucose cannot be compensated. You have to make more glucose. So the glycolysis will be halted, and pyruvate kinase in that way will be regulated in a way that the reaction will move from pyruvate towards the formation of glucose. Conclusion: so the regulation of metabolism specifically, generally, and regulation of glycolysis specifically is an intricate process. It involves many moieties, many molecules, and they have to interplay with each other. They have to work together in coherence to really keep the process of metabolism to move coherently. Some points here, some questions you have to ask yourself. What is meant by feed forward regulation? It must not be difficult for you now. What is meant by if a reaction is regulated by a product? इसका मतलब क्या है कि एक रिएक्शन को एक प्रोडक्ट रेगुलेट कर रही है. आप अपनी डेली ईटिंग को, आप रोटी खाते हो, पानी पीते हो, हाउ यू कैन रिलेट इट विद द रेगुलेशन ऑफ द प्रोसेस ऑफ ग्लाइकोलिसिस. इस चीज की सिग्निफिकेंस क्या है? यू हैव सीन दैट in both glycolysis 1 and glycolysis 2 topics, you have seen that there were certain reactions which were reversible, certain reactions were irreversible. यानी कुछ रिवर्सिबल थे, आगे की तरफ जाते थे, दोनों तरफ जाते थे, कुछ इरिवर्सिबल थे, यानी कि बस आगे की तरफ चल के पीछे की तरफ नहीं जाएंगे. इसकी सिग्निफिकेंस क्या है? इसका मतलब क्या है? ये अहम क्वेश्चन है, तो ढूंढिए इनके आंसर.